Exploring Catalyse activity. Hello. My name is Vito. Today we're going to perform two experiments that are designed to observe how environmental factors such as temperature and pH affect the activity of enzymes, specifically catalase, which is present in potatoes and yeast. Catalase breaks down hydrogen peroxide into water and oxygen, which leads to the formation of foam when dish soap is added. When hydrogen peroxide breaks down into oxygen and water, you will see the release of oxygen in the formation of bubbles, or foam. By observing and comparing the visible changes that occur with each sample solution, we will better visualize the enzyme activity that happens in the body. For both experiments we will use the following materials: dish soap, hydrogen peroxide, a measuring cup, tablespoons, teaspoons, a strainer, a permanent marker, paper towels, 5 flasks or disposable transparent plastic cups, 1 package of dry yeast, 1 medium potato, tap water, boiling water and a freezer, an acid, like vinegar or lemon juice, an alkali, like sodium carbonate, and optionally, some food coloring. Experiment number 1. Testing the catalase activity. In this experiment we will see how changes in the temperature and pH can affect the enzyme activity. To achieve this, we will use small pieces of potato and place them into different conditions. In each situation we will see how the enzyme activity is affected by assessing the amount of foam released. We first label our flasks with the following tags. Control. Boiling water. Frozen. Acid. And finally, alkali. Then we add some tap water to the control flask. We pour half a cup of boiling water in the boiling water flask. Then, we add half a cup of tap water and 2 tablespoons of sodium carbonate inside our alkali flask. And we stir a little. Finally, we add inside our acid flask half a cup of vinegar. We cut the potato in little dices of about 1 centimeter by side. And then we place 3 cubes of potato inside each of the flasks. We leave the potatoes resting in their treatment flasks for 10 minutes. And put the frozen flask with the potato dices inside the freezer. We now label our experimental flasks with the same names as before. In each flask, we add 20 milliliters of hydrogen peroxide and 1 teaspoon of dish soap. After 10 minutes, we used a little strainer and a fork to grab the potatoes, while removing the solution of each treatment flask. We then place each set of 3 potato cubes from the corresponding treatment flask into their related, experimental flask. After 5 minutes, we can observe that the enzyme activity is highly sensitive to changes in temperature and pH. Optimal conditions, like neutral pH, and a moderate temperature, yield the most foam. While extreme conditions, like boiling, freezing, highly acidic or alkaline environments, result in a decreased enzyme activity, and therefore less foam production. Experiment number 2. Testing catalase activity. In this experiment, we will simulate the function of catalase enzymes in the body, and how the amount of substrate or product might affect the rate of reaction or the amount of product released. We will use hydrogen peroxide and yeast, which contains catalase, to experiment with samples containing different concentrations of hydrogen peroxide. As the hydrogen peroxide reacts with the catalase, it breaks down into oxygen and water, and we will observe the release of oxygen as bubbles or foam. By observing and comparing the visible changes that occur with each sample solution, we will visualize the enzyme activity that happens in the body of a living organism. We took a flask to dissolve the dry yeast in about 1/2 cup of warm tap water, and let the dissolved yeast rest for at least 5 minutes. Then, we label our flasks from 1 to 4. To all the labeled flasks, we added 1 teaspoon of dish soap. Now, depending on the glass, we add different amount of hydrogen peroxide solution. Nothing on the first one. 1/2 tablespoon on the second one. 1 tablespoon on the third one. And finally, 2 tablespoons on the fourth one. We also added some food coloring, to improve the visualization of the experiment results. Now, we took the yeast solution and added a tablespoon to each cup. In flask number 1, after adding 1 tablespoon of the dissolved yeast solution, there is no visible reaction happening. Since flask number 1 contains no hydrogen peroxide, there is no substrate for the catalase enzyme in the yeast to react with, resulting in no foam or other reaction products being formed. In flask number 2, after adding the yeast solution, we see some foam formation and bubbling. The catalase in the yeast reacts with 1/2 tablespoon of hydrogen peroxide, breaking it down into water and oxygen. The oxygen gas produces foam, indicating that a reaction is taking place. In flask number 3, after adding the yeast solution, we see more foam formation compared to flask number 2. With 1 tablespoon of hydrogen peroxide, there is more substrate for the catalase enzyme to react with, resulting in more foam production than in flask number 2. In flask number 4, after adding the yeast solution, there is even more foam formation compared to flasks number 2 and 3. This is because flask number 4 contains the highest amount of hydrogen peroxide, 2 tablespoons. Therefore, the reaction produces the most foam, indicating a higher amount of oxygen being released due to the increased amount of substrate for the catalase enzyme. Finally, when all 4 flasks are placed next to each other, we can observe that flask number 1 has no foam, flask number 2 has some foam, flask number 3 has more foam, and flask number 4 has the most foam. Why? This happened because the enzymatic reaction took place in all flasks that contained hydrogen peroxide, flasks number 2, 3, and 4. The differences in foam production are due to the varying amounts of hydrogen peroxide available for the catalase enzyme to break down. The more hydrogen peroxide present, the more oxygen is released, resulting in more foam. Flask number 1 had no hydrogen peroxide, so no reaction occurred. I hope you have enjoyed these experiments. Thanks for watching.